No problem. I'll take it away. So I'm, I guess, at the coalface of a lot of Wolbachia releases, and as Christian mentioned, of course, this is expanding to new sites all the time. And I did some of the original. In fact, I probably ran the original Wolbachia release many years ago, about, gosh, 12 years ago, and I continue to be involved in several programs. So what I want to do is just take you through some of the logistics of how one does these programs and just point out, where modeling has been used. Statistical approaches, modeling approaches have been used in various areas as one goes through one of these programs. That's the aim here. These are just some of the people in my lab. Some of them actually do run models, believe it or not, but there are also people here that do a lot of the practical work. So it sounds like you've been talking about Wolbachia, which is great, and I'm not quite sure. Yeah, that's it. So when you do an actual release program, you have to consider a number of components. And the first thing to do is to obviously think about the strain developments, but also very much understanding the environment in which you're going to do a release. Every environment is different. So for instance, as Christian said, we're going to start looking at Paraguay, and the Paraguayan environment is different to the Australian environment where we started our releases, and it's also different to other places we have worked. So for instance, we also work in Saudi Arabia where it's very hot and very dry. And I've just come from Singapore where it's obviously very mild all year round and there's an awful lot of rainfall. So when you run a release program, you have to start worrying about these things because you want it to be successful. And of course, one of the challenges you have in a release program is that you have to engage the public and you have to engage the regulators and you have to engage the municipality people because you have to sort of keep them happy. So that's always a challenge. You then also have a rearing team that makes the mosquitoes, a release team that gets the mosquitoes out in the field. And of course, there is a very important component here, which is the data science and the mapping and the monitoring as well. So you collect a lot of information. And of course, that also means that it's a very rich environment from the point of view of modeling. And of course, you also then have an advisory team to help you along the way. So that's roughly what we set up when we do one of these big release programs. So the background information is where I'll start. And I've just indicated a few things in yellow here, just marked a few things in yellow, which is really the ones that might be relevant to you because, we do analysis and modeling right from the start, even when we're collecting background information. So you've obviously got to select your sites that you want to work with, and that's often based on the number of dengue cases, the amount of disease that you have in a site. You have to collect a lot of information about the mosquitoes themselves, and you also have to engage, obviously, the public and the local authorities and the willingness for them to do it, which, can be quite a challenge. We generally collect quite a lot of molecular data before we start, and we also look at insecticide resistance, and we do that through bioassays and molecular data. But again, we tend to also model insecticide resistance patterns and how they might change as a consequence of Wolbachia releases. And then, of course, at that stage, before you start releasing, you have to actually plan the best way to do your releases. What's the planning of the optimal design to do your releases? And these are the sorts of things that go into it. But that's, you really need some sort of decision support to maximize the outcomes you're going to get. I'm just going to give you a couple of slides, just giving you some examples of this in a program, particularly the one that we're running in Saudi Arabia at the moment, because that's a bit further along than the program that Paraguay will be running later. And this is, again, very much a program where we act as the advisors and it's all locally run, and that's the way that we tend to work. So here's an example of some entomological data that we've collected. So we have a couple of target areas, one's called Salama and one's called Safa. This is in Jeddah in Saudi Arabia. So that's on the coast, and they have quite a big mosquito and dengue problem there. And what we typically do is set up quite a dense network of traps. So each one of those circles is a trap. And then we monitor those traps for a period to see the distribution of the mosquitoes in these traps. So, part of the reason you're doing this is you're trying to get a feel for how many mosquitoes around there, which then informs your release rate. But also you want to look at the spatial distribution of the mosquitoes to see if you have concentration somewhere. So in this particular case, we had a specific question about the basements of houses. Do we find mosquitoes in the houses? Do we find mosquitoes in the basements of houses or outside? And the idea was that the locals thought that basements were very important. So we basically set about putting out traps and comparing basement estimates with the other locations. And if you look at the estimates that we got from these Bayesian models, it's pretty much showing that the estimate of, minus 0.12 has a very large confidence interval or credible confidence interval of, expands zero. So there's no indication here that the basements are particular hotspots of mosquitoes, which is very important for us moving forward when we do our releases. So these are the sorts of specific questions you're going to address in your entomological data. And then, of course, we also are interested in hotspots, spatial hotspots. So here we have an example of the Salomar site. And so what we're doing here is we're taking all that entomological information and we're analyzing it spatially. And, we typically use kriging for that. And if you look at that across time, you can see that there's a couple of measures here, mosquito presence and eggs per felt. And you can see that you do get some hotspots, but the hotspots actually shift over time. So eggs per felt shows a hotspot on the left-hand side, which then moves down. And that's roughly matched by the presence or absence of mosquitoes in the traps that we collect. But this tells us that at certain times of the year, when we start releasing, we may release at higher densities in these hotspot areas. So again, this is very useful information moving forward. And these are just some of the estimates in different locations for these hotspots and how far apart they are, which again is, of course, useful from a release point of view. So what we also do is we collect information on mitochondrial DNA, and mitochondrial DNA is an incredibly important tool for monitoring what happens to the mosquitoes across time when things go wrong. So if your releases don't work very well, the mitochondrial DNA can actually provide a lot of interesting information. And I've just been talking about mitochondrial DNA problems in Singapore, which shows very much how this is very useful. So this particular top situation is basically the incompatibility situation. This is the one you're familiar with, where you have Wolbachia-free mating with Wolbachia-infected. So, of course, you have this situation in the bottom left where the males that carry the Wolbachia sterilize the females. So, that's the classic incompatibility that allows the mitochondria, the Wolbachia to spread. But of course, what happens is that you don't only get the spread of the Wolbachia, but you also get the spread of the mitochondria. And that, of course, is critically important because if you get a dissociation between the Wolbachia and the mitochondria, it gives you an idea about, a situation where you might be encountering problems. So there's two of these problems are indicated in the bottom. One of these problems you might have is something called maternal leakage. So if the Wolbachia-infected females start producing a lot of uninfected offspring, then you'll get a dissociation between the mitochondrial type and the Wolbachia infection type. So when you see that happening, then you know there's an association happening as indicated in the top right quadrant there. You're getting these uninfected individuals that carry the original Wolbachia mitochondrial type, and you know that's going to be a problem. On the other hand, you can also look for migration. Again, in Singapore, we had a big migration event, which really became quite messy. We've had one in Malaysia as well. And you can detect that by, again, just tracing the mitochondria at the same time. So very important information for us to collect as background. And here's an example of the Saudi situation. It's very complicated mitochondria. So the green ones are the Saudi mitochondria that you find in the natural population. It's very complicated. But in South America, it should be a lot simpler. We should have only one clade of mitochondria popping out. And if you have an introduced mitochondria along the Wolbachia, you can then track those problems that I mentioned before. So we also use genomics these days and modeling to actually understand the movement of the mosquitoes in a very small space. So Tom Schmidt in my lab has done a lot of work on this close-kin diet analysis. So what we do is we sequence mosquitoes from a small area to see how far they move. So what you're really doing is you're looking for relatives to see which mosquitoes produce relatives over a certain distance. And you look at first-generation relatives and second-generation relatives and third-generation relatives. And that can provide a good indication of how much movement of the Wolbachia you expect once you do your releases. So here's an example in Saudi Arabia, and we were particularly interested in this big black line down the bottom, which is a big highway. The other one's a small highway. And we then look for full sibs and half sibs and first cousins. And we ask the question, how often do they cross the road? So if a road is a barrier, you don't expect to see a lot of full sibs crossing the road. You don't expect those linkages between those red squares on the left-hand side crossing a road. On the other hand, if you have yellow squares that occasionally cross, then it might indicate that you do get some movement across that road after a couple of generations. So it allows us to very powerfully see how far the mosquitoes go once you start doing releases. And in this case, the big road acted as a bit of a barrier, but the small road did not. So that's all genomic data, but then use modeling to actually understand what's happening. So out of that sort of information, we then typically make decisions about whether we go for suppression or we go for replacement. Replacement is much more commonly used these days because it's easier, it's much cheaper. And often, by the time you collect the background information, you realize that the suppression is going to be a bit complicated. But there is a large suppression program in Singapore that covers 25% of the island now, and that's being quite successful. It is possible, but you have to spend a lot of money and have a lot of resources. So background information, critically important, and of course, then you can actually start building your models. Now, the simplest models for replacement go way back to 1959, and that's a paper by Kaspari and Watson, and just a simple deterministic model. And, it looked a little bit like this equation here, equation 2, and it basically says the probability of Wolbachia changing from generation 1 to generation 2 is a consequence of a few key parameters. So you've got this mu, which is the transmission rate, the maternal leakage rate. You've got H, which is the hatch ratio of the eggs, so that's the incompatibility rate. And you have F, which is the fitness. So those are the key parameters that we typically look at in these early deterministic models. And these are the models that, like I said, started with Kaspari and Watson. And Michael Freley has worked on a lot of these models as well. And we've actually tested them biologically, and they actually work surprisingly well. But, they are very simple models. They work in a lab context, and, they are based on recursions. And we use recursions because the fitness of the Wolbachia under incompatibility is frequency dependent. It tends to have Wolbachia tends to have a much lower fitness when it's at a low frequency, and it builds up as you get more infected individuals then into the population. And then, of course, eventually it starts to really take off. So here's an example of the sort of data you can generate in the lab to support those sorts of models. This is actually in mosquitoes. This is a paper from 2016. And here we have the LB infection of Wolbachia. So we set up a bunch of different cages, and what we do is we start at a particular point. So we predicted the invasion point. So that's really based on the balance between the fitness cost of Wolbachia, the hatch rate, the incompatibility, and of course this leakage rate, this maternal leakage rate, this mu, and that basically can predict what should happen. So we predicted this one should be around 18% or so, and as you can see, works pretty well, right? The black lines go up when you're above that unstable point. The Wolbachia takes off. When you're below that point, the Wolbachia doesn't do much. There's too much of a fitness cost, a bit of leakage, and potentially even a lower hatch rate that can affect the Wolbachia invasion. So this type of information was used in field when we first started doing the releases. How high do we have to get in the Wolbachia to actually kick off this invasion process? And it's simplistic, but it actually does have some predictive power. So I'll go over these very quickly because they're not so relevant to you, but obviously when you do a release program, you have to read in mosquitoes. This is a very small release lab, which we set up back in 2010 to actually help the releases in Australia. Some of the first releases are very small, nothing automated, very simple. And then, of course, you can go to much fancier systems. So this is actually a factory, one of the 2 factories in Singapore where they're producing millions of mosquitoes. In this case, it's a suppression program, and they are at about 5 million mosquitoes. Male mosquitoes, actually, is at 10 million mosquitoes a week in this production facility at the moment. So it gives you a bit of an idea about the scale. So this one here produced about 20,000. This is going up to 10 million a week. So we've also seen massive developments in releases. Typically, when we start releases, the technology is very simple. So this is some shots from releases happening in Saudi Arabia at the moment. Very simple. You have the mosquitoes in a cup. You pull off the screen and the mosquitoes fly out. So we typically have about 80 to 100 mosquitoes in a cup like that. So that's one extreme, and then of course you go to the other extreme. So this is the debug system that rarely used, where they shoot mosquitoes out automatically from a van. This was used in Australia. It's also being used in Singapore at the moment. And it was also used in California and Fresno. So low technology to high technology. Data management is critical in this exercise. This is something you need to set up. We tend to use barcoding systems a huge amount to track, where the mosquitoes are released, also to actually then use them to put traps out when you're measuring the Wolbachia, and of course then also looking to see what happens to the frequencies over time. So barcodes, traps, barcodes, release points, and that allows you to actually really track the invasion of the mosquito very, very accurately. And then, of course, you can do your spatial analysis and what have you. So what's happened over time? Well, there are many, many releases, but Christian was particularly interested in the complications that we might encounter as you go out to Paraguay and other places in South America. And I'm going to focus on those. So this is the early release that we did back in 2010, 2011. And the green line is the frequency of Wolbachia. Goes up very effectively. The gray bars indicate the releases, and you can see that we're releasing very low numbers in this case. That's why we only had to produce 20,000 a week. But nevertheless, the Wolbachia in this particular area went in pretty well. And it went up to close to 100%. And that's actually still at that frequency after all that very, very long period. So that's an example of simple things, goes well, walk away from it, you forget about it. And in this particular case, we walked away from it. We stopped releasing, as you can see, when the Wolbachia was at a frequency of at least had to 80%, I think, then we stopped 70 to 80%. Because we knew that was way above the unstable point, which in this case was predicted to be around 20 to 30%. So we wanted to play it safe, and it paid dividends. So here's some releases with a different Wolbachia strain that we've been involved with in Malaysia. And this shows you the complexities you might encounter. So in a sense, this is simple, this is complicated. And The first site is pretty much very simple. So this is a place called Mintari Court. It's in Kuala Lumpur. And, we started releasing for a fairly short period, as indicated by that grey box. Went up, stayed up, and it's still up. And, now 2023, it's as high as it was back in 2018. So very effective, very easy. In the second one, we have a situation that's a bit more complicated. Went up very quickly. We didn't have to do too many releases. Mosquito went up, but then they crashed again, right? So, there's many explanations for that, which we'll come to in just a sec. But then we started releasing, went up quite high, and then it's stayed stable since that time. We stopped releasing in 2018, the end of 2018 there. It's been stable ever since. You've also got situations where sometimes it takes a while for the mosquitoes to go in, and sometimes you get these big dips. This is the commercial sensor here. You can see this dip. And we wanted to make sure the mosquitoes were stable, and they became stable after a while, and of course everything's been fine. This third example is interesting because we never got it in. So, this area AU2, you can see that the mosquitoes go up, down, up, down, and we went to a low-level releases every 2 weeks. So you can see those hatched bars on the right-hand side here, and it still keeps going up, still keeps going down. So it's a fascinating situation where you simply cannot get the Wolbachia up. Now, this is not a unique experience. Anyone that tells you that Wolbachia invasions are always straightforward, don't believe them. So here's some work that's been done in Rio with WML by the FIA Cruise group. And, again, if you look at this paper, you can see it goes up and, it does quite well, but then it starts to go down. But then it seems to go in waves. And you can see the gray bars here that are releasing the mosquitoes, right? So the mosquitoes are obviously quite variable in this case, and you're not really getting an invasion. So it never really works in this particular case, which is kind of interesting. So Rio is actually quite a challenging environment, and it's been quite difficult getting stable invasion by the FIA Cruise group, despite the fact that they've put a huge amount of work into it. So once you obviously get all this detailed spatial information, it's all barcoded. The traps go out, the traps come back. The frequency of Wolbachia at every single point, and then you can have a lot of fun, right? You can basically model away and analyze away, and you can produce some beautiful graphs. So this is a recent one from an area that we're releasing in Jeddah, Saudi Arabia. And you can see that in this particular case, we have a pretty high frequency of Wolbachia all through. There's a bit of a weak point at the top, but, surprisingly strong down the bottom. So not too bad. I mean, not much heterogeneity here. And we have release here at a fairly even level. But there's also situations where we've had trouble invading at the local spatial level, and I've got them to bump out more mosquitoes at that point. And that's usually managed to overcome it if you're in this gradual increase situation. That's the sort of, this one's just kriging analysis. I think it was done on ArcGIS, if I recall. So that, that's a bit of a snapshot, but over the years, as we've done releases, we realized that, things are a bit complicated. And you've obviously got to think about the environment. You've got to think about the actual, you've got to think about the actual public engagement. You've got to think about costs of the infection. You've got to think about the local situation in terms of insecticide applications. So there's a whole lot of environmental factors that you need to think about. And you've also got to think about other things like disease pressures and also the mosquitoes themselves. So, this is quite a complicated picture of what we've learned. And all of those factors influence the spread and persistence of the Wolbachia into the population, or if you're doing suppression, you encounter the same sorts of problems. And of course, you've also got what might happen down the bottom. So I'm just going to go back to this one, because of course things can evolve, things can change. It's not going to be static. So this is something that we also worry about, but this is something that we worry about the long-term rather than the short-term, because of course, both the host can evolve, the virus can evolve, and the Wolbachia can evolve, and that can modify things again. So I'll keep it simple for now and just focus on those top things. So what we can call one the environment. We've got the host background, we have the Wolbachia variant, and there are several variants available. There's WML, there's WLB, but there's also different types of WLB and different types of WML these days. We have these operational issues, sorry, it's jumping again. And of course, we also have the mosquito population itself. So this gives you a bit of an idea about the issues that arise and what you might need to incorporate when you're setting up different models. And obviously this goes way beyond those earlier deterministic models that were used to explain the frequency changes of Wolbachia in laboratory populations. So I'm just going to cover a couple of these issues just to show you what can happen. So here we have a dissected female reproductive tissue, and that thing on the right-hand side has two little wings, and those wings are actually the ovaries of a mosquito. So the mosquito on the left has no ovaries. In other words, it's going to produce 0 eggs, and the one on the right is normal. So where does it come from? You know, what's going on here? So it turns out that this is all to do with temperature. This is all to do with Wolbachia interaction with temperature. So what can happen is that when you have a mosquito population, sometimes if you have unfavorable conditions due to temperature or also due to dryness, then the eggs have to stay in a quiescent state for a long period of time. So if it's hot and if it's dry, then the eggs often just stay there and they can hatch only when things cool down or when the rainfall comes. So what you have is you can look at the hatch proportion of these eggs across different storage duration periods. And this can be quite dramatic in terms of its effects on hatch rate, particularly when you're Wolbachia infected. So under A, if you look at this A graph on the left-hand side, you can see that the gray line indicates what happens when you're not infected by Wolbachia. There is actually one situation where a genetic background effect also kicks in, which is that gray dotted line. So normally Wolbachia looks like the top one, but in some backgrounds it looks like this dotted one. But then you look at this LB, this blue line, and you can see that the situation is quite different. So with LB, you can see there's this sinking effect as the eggs have to stay in a quiescent state for longer. So what this effectively means is that if the hot conditions or the dry conditions mean that the eggs don't hatch for a while, then you can lose the Wolbachia. There can be a massive fitness cost, which is climate related. Now, you not only affect the hatch rate of the eggs, but you also affect the fertility of the females that come out of those eggs. And in some ways, this effect is even more dramatic. If you look at those two blue lines, you can see that they go up as the egg duration period increases. And the gray line indicates the uninfected ones, and they're perfectly happy. So this is a climate-related or a condition-related cost of Wolbachia. And of course, if you think back to those deterministic models that we have, you know, this is much, much more complicated. So the environment can dictate the fitness cost, unfortunately. The other intriguing thing that can happen is that you can also get an effect on the leakage of the Wolbachia itself. So I mentioned that leakage was a key condition. And what can actually happen is that the sexes can start to lose Wolbachia under hot conditions. So at the bottom here, you can see a graph that there's a cycle between 26 degrees and 37 degrees. So that's a cycle that reflects what can happen under quite a hot set of conditions. And what happens is that you actually can induce incompatibility. So this is an infected line, female times male, WML times WML, it's one of the Wolbachia ones, and it should be compatible. And yet, when you put these, when you rear these larvae under this cyclical condition, then you can get incompatibility. So the males start becoming incompatible to females because the Wolbachia starts to drop out of the females in particular. So you get an incompatible situation because of a change in the density of the Wolbachia. So again, from a modelling point of view, I'm just highlighting the fact that this is another cost of your Wolbachia. So Wolbachia is normally spreading my incompatibility, but here it's actually generating incompatibility with itself. And, there's data from Vietnam that suggests that this is actually quite the case. So very interesting. And of course, people have jumped on this sort of thing and said, oh, well, maybe under climate change, let's model this and see if Wolbachia will become unstable. And this paper here is an example of some of the challenge you have in this modeling. And we've actually, because what they did was they took a whole lot of our data, our temperature data, and said, well, let's try and predict what will happen in the future. And we can then say, well, climate change isn't going to be extreme enough to cause these sorts of problems to knock Wolbachia out in the future in cases where you've had an invasion. But, this paper makes a lot of assumptions that I actually disagree with, and we have written a critique of this, but that's not really the point. The point is that, you can then model what happens under climate change in the future. You're just going to make sure that the model you're using is an accurate representation of what's actually happening. So I like the approach, but I'm not quite sure about the assumptions that underlie it. So one of the other things that we are doing to overcome these sorts of issues is to basically say, look, things can go wrong occasionally. And so maybe sometimes Wolbachia needs a bit of a helping hand. And so what we're also doing, particularly in Saudi Arabia, is to look at useful adjuncts to the Wolbachia program. So if we do get a sudden, burst of Wolbachia, burst of mosquitoes, and high temperatures and dry conditions mean that you get these sorts of egg hatch rate problems and egg sterility problems, what can you do? So this is sort of our backup strategy that we started working a lot on. And this is actually a trap. It's called an in-situ trap. And it's very effective at bringing down the mosquito numbers when you are actually running into one of these issues, when you get a sudden increase in mosquito numbers. And then, you might go over the top and get some more releases happening. So this is something that I think we just need to be prepared for in the future, particularly under climate change and the massive variability that climate change is bringing around the world, including in Wolbachia release areas. There are ways of doing that, but again, I think there are potentially some quite useful models that could be done in terms of when you kick in these sorts of strategies, when you decide that things are really going downhill and you need to do something about it. So, what I've done, I've sort of covered some of the issues, shown some of the complexities, and pointed out, hopefully, a little bit about where modeling becomes useful. But of course, ultimately, what we also want to do is we want to look at the impacts on dengue. And, this is kind of an interesting area that's getting quite hot. And there's quite a lot of disagreement among people that are setting up the epidemiological models. So obviously, the traditional approach you would use is you would release your Wolbachia in a randomized control trial of some sort. And you might have arms if you're trying to match the area, the treatment and the control area, and have a lot of arms. But and Wolbachia is quite difficult to do. I mean, there was one randomized control trial set up in Indonesia, cost a huge amount of money, and of course, dengue is one of those diseases that only comes on occasionally. So you could, in Singapore, they also set up an RCT, and they're running into trouble because this year was a very low year for dengue in Singapore. So, they can't really collect that much useful data. So RCTs are possible. They are a problem when it comes to mosquitoes and Wolbachia because to get stable Wolbachia in one area and have a control area that doesn't have Wolbachia, you need quite a large area. And you can get spillover of the Wolbachia, so the Wolbachia might start spreading out if it's very successful. In some areas, it just keeps spreading out, so you get spillover as well. And, you can make sure your control area is a bit, as far away, but if you've only got a limited area, that can be quite difficult. That's one problem. Replication numbers is also a problem. You find yourself soon, if you talk about 5, 6, or 10 replicates as this thing may require, then it's very difficult to do at the same time unless you've got a giant factory set up. So RCTs are a problem. And one of the other things that can happen is you can get a lot of political pressure to abandon an RCT. So in Malaysia, we were running an RCT, and so we had a lot of sites which were dengue hotspots. We randomized it, even got the politicians to randomize them, and we said, well, these are going to be treatment sites and these are going to be control sites. Picked the treatment sites, started releasing the Wolbachia, and then of course, all of a sudden there was a panic because one of the control sites had a huge blip of dengue, and the health department said, right, we have to release Wolbachia. You know, you've shown that it works. We have to get the Wolbachia in there. And, you know, so eventually, of course, we had these sorts of issues and a lot of things, and we just had to abandon the RCT. So that's a problem that can happen. So these days, there's quite a lot of work on trying to look at alternatives which have more power than RCTs. So there's a synthetic control trial approach, where you look at periods of before and after, and you try and look at all the variables that can bring those together. That's more observational. And of course, what you can also do is just think, well, it's all too difficult, and I'm just going to do a site matching approach. So I'll have a bunch of release sites, I'll have some matching sites, control sites, and my model is simply going to have a treatment effect, so before and after effects, before treatment, after treatment, look at the incidence of dengue, and just match them up. And that might even be done retrospectively, which one might have to do in operational sense. So, of course, you know, these are the sorts of issues that I'm just talking about. So size of release areas, similarity across control release areas if you want to use arms. Also, the quality of the dengue data can be quite variable. And the dengue monitoring, it has to be fairly even, obviously, across the period you're looking at. The time period, you need several years of data. You know, we typically work with 5 years of data. And as I said, the best laid plans can go wrong because of the political considerations. And I think, you know, one of the things about Wolbachia replacement is that it can be shown, you know, it produces results quite quickly, even though they scientifically are not rigorous. And then, of course, it leads to excitement in the community. So in some of the work I've been involved with, we've had to take shortcuts. So this was the paper that we produced out of Malaysia in current biology, where we estimated a decrease in Wolbachia of about 40%, and that involved a match control site, release site approach. And then we had a couple of people, Nick Goldings from Nick Goldings' group, apply a Bayesian model to that to estimate the decrease in dengue and the critical intervals. So here's some, an example of the sort of output from that. And you can see that the model predicted a dengue reduction of 40%, 40.3%, quite wide confidence intervals. It's the nature of the dengue data. And of course, you can see visually that things look pretty good, right? You're getting dengue and then you start releasing the blue area and it goes down. But of course, we've got a whole lot of mesh control sites, and some of those also show these sorts of effects. Now, one of the advantages, of course, is you can keep adding data, right? You can just build up a much larger database. So in KL, in Kuala Lumpur, where things have become operationalized, we've now got actually 40 release sites. And we've also got a lot of control sites. So in this study, we have 20 release sites with enough dengue data, and then we have 6 control sites. And they're all these high-rise sites. Just give you a couple of examples. Sometimes they go 20 stories high, and sometimes they go about 5 stories high above some commercial premises. So these are just typical buildings that you get in Kuala Lumpur. And they're all dengue hotspots, right? So these are all areas where you get a lot of dengue. And so, that's what we did. And so just gives you an example of the heterogeneity you get in these Wolbachia releases. Sometimes they jump up and down and then it becomes stable. Sometimes they go up very quickly and they're stable. Sometimes they go up and then they come down and then they go up again. And sometimes they go up, but it just takes a long time, right? You have to release for months and months, and gradually it goes up and then it stays up. So, there's obviously totally different dynamics in these situations that are happening. And they're a little bit hard to predict, believe it or not. I can't go into an area and say, hey, this is what's going to happen. And all those factors I've been talking about sort of drive that. And of course, some of the breeding sites have frequency dependence built in. So, a mosquito larva can take a week to come through, and a mosquito larva in a dense situation might take several weeks to come through to actually develop, particularly if there's not much food around. So you can take a water tank, the water tank doesn't have much food, and it's all someone throws a chicken or something in there by mistake, and then the larva will take off. So those are the sorts of challenges you face. And of course, again, in Malaysia, we had the odd situation where we're not getting much in. But, you accumulate the data, you run these Bayesian models, and this just shows you how well they work. They're pretty good at predicting. So you've got the colored bars and the gray bars. One's the data, the other one's the actual predicted data, and the green area shows you a couple of release sites we were talking about after the releases. And, so here's the whole lot, and obviously, as you on the right-hand side is the paper we just submitted. Also, a lot of people, typically that's what happens. You get these little blue areas, but importantly, your predictions become better and better, right? That's the point. And so now we have Wolbachia frequency, 82% on average, 56 to 96. Some areas have been very well invaded. But that initial estimate of 40% is now gone up to 62%. But importantly, you can see there's a 95% interval being really, really tight now, right? 50 to 72, as opposed to 5 to 66. So you can accumulate this information. I think it's one of the joys of these sorts of Wolbachia releases when you've got matched control sites. And we also showed, incidentally, in this case, that it's critically dependent on the Wolbachia frequency. Once you get a Wolbachia in, then you get pretty good blockage. So all the way from 42 to 73%. So, so that makes us pretty happy. And I think that's why when you start your releases, you have to consider it as an ongoing project. You learn, you adapt, and you keep going. And eventually you get to some pretty nice results. So what is our lesson from that? High Wolbachia, stronger reduction. Interestingly enough, we didn't see a lot of effective site characteristics, which will surprise us. So you can have buildings look the same, but they still have different breeding sites. And like I said, it's pretty hard to predict. And of course, these were hotspots, quite small areas. And of course, a lot of the dengue acquisition is outside those areas, people bringing it in. So that counts as a dengue case in the area, but it's not local transmission, which of course is more important. And yeah, you get a few weird things happening. We had a construction site being built and totally threw up off one of the sites. But, again, these sorts of things happen. So, the data looks pretty good for Wolbachia when you start adding a lot of studies together. This is from different countries. So it can work, but the local situation is a little bit complicated. So I just wanted to give you a bit of a snapshot of what we've done. And obviously this is funded by many people. This is a bit of a snapshot of my current funding. But, it's an interesting exercise, and hopefully I've highlighted a few places where I think modeling can make contributions. Trick is going to be to look at the local situation and be able to produce useful models for that situation.
Thank you, Ary. So we're here. We are some 20 colleagues from several areas, and professors and students as well, biologists. So maybe there is some question about. Let's start.
So thank you for the talk. My name is Marcelo. And I wanted to know, given that there is a couple of new vaccines against dengue that are just coming out and were tested in the population, what do you think that are the perspective of this sort of works? What do you think about the idea of just proposing fighting dengue with mosquitoes instead of vaccinating the population?
Look, my answer to that's pretty simple. I always think that you just need a bunch of different approaches, right? I mean, the thing about dengue being combated by Wolbachia and dengue being combated by vaccines, those two can work together, right? Don't forget, a lot of vaccines have come out over the years. A lot of them have proven to be problematical. I mean, it's great to hear there's still new ones on the horizon. As you say, there's a couple of new ones being released at the moment. They may turn out to be quite good, and they may work in some situations, not in other situations. But, you know, you need multiple approaches to get rid of dengue, right? I mean, I always view this as very much a case of one approach not being particularly incompatible with another approach. So, yeah, go for it. Do your vaccine trials, but in your vaccine trial situations, do them away from your Wolbachia. But eventually, of course, it may be that you use both modalities to push down dengue. And that's also why I highlighted the whole, you know, propoxivin and 2-care approach. Because I think, again, that can work very well with Wolbachia. That's, you know, it's a totally different approach that actually can be integrated. So thank you for the question. But I think, you know, the important thing is not to think about silver bullets, but to think about situations where one approach complements another approach. But while you're testing it, obviously, you need to make sure that you're only using one approach. And we've encountered that in Malaysia where some of our release sites were then being used by another route for doing something totally different. And didn't go down particularly well, became problematical. It wasn't vaccines in that case, but it was repellents that became a problem for us in one place. Yeah.
Okay, thanks.
Yeah. Hello?
Yeah.
Hi, my name is Paula. I want to know how the validation stage in the project, in the research is performed.
You mean the validation stage? Well, the dengue validation stage or the entomological validation stage? There's two validation stages, right? I mean, on the one validation is that you're getting it all back here into a high frequency and it's stable, right? Without doing more releases. So that's certainly one way of validating that the thing is working. But the second one is obviously the reduction in dengue, right? And that's why I was giving you a bit of a snapshot of the situation in Malaysia and those hotspot trials, where, you know, I think the validation is pretty good, right? Because we've seen a real live reduction in dengue of over 60% now. And we've also, you know, we're not the only ones, of course. That's with WLB. With WML Indonesia, they got a very good result. They got a reduction of about 80%. And in Australia, of course, we've got rid of dengue entirely, local transmission. So I think, you know, I mean, I think the approach is being validated and across different trials, you know, if you put them all together, like I did in that last graph, as long as you get the Wolbachia at a high frequency, I think that looks pretty good. And of course, you know, like vaccines, we're also improving things all the time. So we have a strain at the moment, a WML strain that we think is a lot better than the original WML strain, right? And that might be able to match the old B strain in some situations in terms of heat resistance. And that is just an absolutely superb blocker, this new one of chikungunya and Zika as well as dengue. Yeah. Lots on the horizon. At the moment, the validation, I think, looks surprisingly good. Stability looks surprisingly good too, actually. So, yeah.
Thank you.
Is that what you meant? I mean, that's how I see validation, right?
Okay, thank you.
Yeah.
Is it done? I understand that the temperature is a problem, right? It's to say that for the WML, it can, how do you say, be incompatible in some sense that they have the Wolbachia and they avoid the Wolbachia. What happened with the other strain, like Albopictus A, B are the others, and because I start to be concerned about what is going to happen in Paraguay with 45 degrees Celsius or 50 degrees Celsius.
Yeah. No, look, it's interesting. I think it's a very good question. I think, you know, at the moment we have, you know, like I said, there are two strains, but variants of those strains. And I think it's important to recognize that. So if I was releasing in a hot climate, I would preferentially release WLB, right? But of course, we've also created this strain of WML, this WMLM, which is actually quite a lot more heat resistant than the original WML, right? So, I think what we're doing in Saudi Arabia to check that. I mean, you know, Saudi Arabia is one of the hottest places on earth, right? It gets up to 50 degrees. Okay? Now, the mosquitoes are pretty good at avoiding heat, but at the same time being exposed to that. But a combination of heat and dryness can be a problem, particularly through those egg hatch things, right? So there's a bit of unpredictability about which strain will be best. And what we're doing in Saudi Arabia is releasing both. We're releasing WML-M and we're releasing albias at the same time. And the warm-up exercise is 2 areas with one and 2 areas with the other, just randomly selected. And we'll see how we go, right? We'll look at the ones that have the stability in those 2 strains. I mean, to be honest with you, I think if I was doing it, I would, there are more strains under development. I'd love to have 6 strains, right? And I'd like to throw, 2 areas or 3 areas under each of those 6 strains and see which one stands up best. Now, 45 degrees, I mean, where we're working in Jeddah, it's 45 degrees a lot of the time, so that doesn't worry me too much. But in that case, probably LB or the more heat-resistant strain of WML would be my way to go. Yeah.
So, Basi, if you had to do a model, try to understand the behavior of the temperature, what do you think is going to be a good model to understand the problem?
I think what you need.
I mean.
Yeah. So I think what you need to do is you need to understand the local breeding sites. Now, you've got good entomology people in Paraguay. They've got a fairly good handle on the breeding sites, and you can measure. You can obviously measure the temperature. See, my approach is that you collect a lot of information, you feed it into the models, and you keep you fiddle the models till you can predict something useful, right? I like that approach a lot. And in this case, you use the data from the entomologist, you actually collect the information, you're starting your trial releases based on your best guess, and then you see what happens, and then you modify those as you're going along. So that would be my approach. So you consider a range of strains, but you also, at the same time, make sure that you can try and model as best you can what's actually happening. And it's not a bad way to go. It's a bit of an insurance policy in a sense, isn't it? So that's what I'll be doing in Paraguay. Yeah, talking to the entomologists, thinking about it spatially, thinking about the breeding sites, thinking about the temperature in those breeding sites, and it may not be an issue. Right? It may not be an issue if it's 45 degrees. Maybe the breeding sites of mosquitoes where you are is, if it's big bodies of water, it's probably not going to be an issue, right? Yeah. Small bodies of water, problems, yeah.
Hi, I'm Claudia. I was wondering about the figures that you show that you were like trying to guess what was happening someplace it was easy, let's say, to keep the infections and the other one no. And then you make some kind of comment at the end that it looks like the place were similar, or you could not establish any kind of relationship in the sense of the environment. And then I was thinking about how was the population dynamic before the introduction of the Wolbachia, and also, do you think this can be related to people behavior or somehow that it's like not in the same, let's say, helping? Or, I don't know, they change behavior, and it can be the reason why you cannot really guess what is happening. So I was trying to think about what is happening, why it's not working, why we cannot say, okay, this kind of environment, it was good, this not, and we could map, and try to be better in the other time. So I don't know if you have some comments on that. Thank you.
Yeah, look, it's an interesting one. So one of the problems of human behavior is that you never know quite what's going on, right? So I remember we had these SOPs, right, the standard operating procedures for putting out traps, and basically, if we had those 5-story houses that I showed you, we said, look, put the traps, not the traps, the release containers, put the release containers at the ground floor, and then put them on the 3rd floor, right? Ground floor, 3rd floor, ground floor, 3rd floor. So one of the sites we're having trouble getting invasion, I went back and I talked to him, and he said, oh, look, I know my mosquitoes. I said, I just release them to the ground floor. Besides, it's too much work, it's too much work to go up to the 3rd floor. I'm old and tired. I don't want to walk up to the 3rd floor. The point is that we didn't change that, and then all of a sudden we got invasion. That's the human factor, the unpredictable human factors. But you know, I do think that the local entomology people have a lot of information, right? And there's a tendency for us to think about it simplistically when it's quite complicated. So, and you know, but once we can understand the complexities by talking to the right people, I think, to some extent. So one of those areas where we, you know, at flat 7, where it sort of went down and came up again once we started releasing, and then we got it stable, you know, was really because we talked to people and thought about it, and we ended up releasing in a somewhat different type of location. And in that particular case, we're thinking about it too simply. But by communication with the people on the ground, we then realized that we were not hitting the right spots with respect to the mosquitoes. So it's a combination of knowledge, collecting knowledge, but also having excellent data to back up your decisions when you're thinking around that sort of area. But yeah, not straightforward, but there's a lot of knowledge out there, a lot of data you can collect that makes it relevant. So not quite sure if that answered your question, but I think it has to be a two-way flow of information, right, between the scientist and the person on the ground who has the most familiarity with that situation. And then you can achieve surprising amounts of things. Not always, it can still be unexplainable, but sometimes it can become more explainable. Yeah.
It's because we have been reading several papers, and one of them is the Brazilian case that the boulevark in some sense was rejected. In some sense was a failure, one of the experiments, right? So the idea is we are thinking what can be wrong, is to say where we can fail in this process.
Yeah, no, look, absolutely. And, you know, I mean, the initial failure from the small scale releases, and, you know, this was nice work that was done by Rafael de Freitas' group, right, who's no longer involved in the project, but he's a good entomologist, right? He knows the mosquitoes, he does a very good job. And that turned out to be insecticide resistance, right? So it's the background, there's very clear data for that. So the KDR locus is a locus for pyrethroid resistance. That's what you normally get in a spray can. And the genes involved in that in the Brazilian background had a big cost, right? So if you read them in a factory for a few generations, then, you know, you end up producing mosquitoes that don't have those resistance cost genes anymore. And then, of course, you release them in the field, and then there's a lot of fog.
You mean resistant for insecticide, something like that? You mean?
So you need to match, you've got to match the insecticide resistance and the release stock for what's happening in the field. And you cannot, you know, you cannot simply pretend that there's no costs in the insecticide resistance. So if you end up rearing for 6 months, rearing mosquitoes and not introducing new material, and what happens is that insecticide resistance goes down, right? So it's certainly the case that in one well-documented case in Rio, the insecticide resistance genes were responsible for the lack of invasion because, of course, you know, you start fogging, and then, of course, you end up killing all the released mosquitoes. That's a bigger problem. So that fitness effect, you know, that I mentioned, that equation can be massive, right? So the unstable point goes too high, you never get invasion. So that's an issue. So that certainly has happened in Brazil. The other thing about Brazil, of course, is that, I mean, see, I don't know the details of what they did, right? I mean, in situations where I work and I know the details, then I can provide comments. And we measure the costs of resistance. We measure the rate of adaptation to the lab. But, you know, a lot of people do it wrong. So in Singapore, we had, you know, I mean, I said to them, if you're producing mosquitoes, and this is suppressions that I worry about mating, I said, use a big cage in your factory, right? You've got to have a big cage. So the males have to fly, because otherwise you end up selecting for males that just sit there and mate the female. When the female is also sitting there, you lose activity in those cages. So lab adaptation can be critically important. And if you look at big biocontrol programs around the world, they run into problems until they fix things like that, right? So you've got resistance genes, you've got lab adaptation being a problem, you've got not understanding a local context and thinking about these sorts of issues in terms of the you know, the diapause and the quiescence period of the eggs when they're actually out, and the climate of the situation, all of those can go wrong. I don't know what actually happened in Rio, but I know that they did get a successful invasion into Chubia Kanga, which was quite early on. But I suspect they just haven't understood the complexity of the situation they're releasing, and I think that's the big problem. But I've never reviewed that program. A lot of the papers that come out of it don't really talk about complexity, and that worries me. Right, that really worries me. Yeah. Especially when, of course, they're embarking on it. To be honest with you, also, I mean, the final point about the Brazilians, of course, goes back to the strain. Right now, you raised the strain, obviously Christian, and they've all used one strain, right? So. And I would, like I said, if I go to a new environment, I would be releasing 2 or 3 or even more strains at once, see which one goes, and then run with that. Yeah.
Well, we have many questions, but we had to finish. Oh, thank you, Ary, so much, and we are going to be in touch. And thank you, Les. Thanks.
Yeah. Yeah. Well, thanks for inviting me. Great that you guys are here thinking about these sorts of issues, and hopefully, at least I've highlighted how useful these things can be. Right, simple models to complicated models really can help inform. And hopefully at the end, I managed to demonstrate in Malaysia that if you keep adjusting things, you can come up with some pretty good predictions and you can really improve things. So thanks.
Yeah, this is a journey that we are beginning at the beginning. So thank you very much. Okay,
all right.
see you. Bye-bye. See you.